Welcome everyone to the PhD defense for Wesley Hall. Wes will get to tell you the story arc of what he did for his PhD research, but I get to tell you the story arc of his life, which I really don't know, although there are some experts here and online. Imagine this small rascally kid from a small working class community who's kind of a pain in the ass inside. And had some really great parents who got him outside on a regular basis, poking around at stuff in the mud and the water. And sometimes he'd get squirted and sometimes he'd get pinched. And that made him yell and laugh and poke some more. And I would say that that is something that Wes has embraced through all of his life up till now. And one of the most fun aspects of being out in the field with him is just a sheer joy of being outside and figuring out how things are working by poking at them, which at this point means you do an experiment. Yeah. Okay, so he fished, he crabbed, he dug clams. Later, he worked in the shellfish industry, doing the hard labor of hand sorting product out in all weather and challenging working hours of people whose lives are run by the tides. In the meantime, he played football, coached middle school sports, worked at a bicycle repair shop, kept his relationship stable with his high school sweetheart, who I met for the first time today, despite the fact that Wes has been in the lab for years. And also started the slow metamorphosis into a scholar, which, you know, that metamorphosis in this day and age is maybe not the most comfortable place, but here he is in this metamorphosis. When I met Wes, he had gone from community college to the regional polytechnic and then extended his time there to get a master's degree and had some funding. And Wes agreed to come help out with it, and then lockdown started. Remember that? Yeah. All right, but the research that we were involved in directly addressed information gaps that were identified by local stakeholders about the interactions between this key organism that he's going to tell you about that's actually in his thesis, the burrowing shrimp. You'll learn more. And other parts of the system. There were serious information gaps because people could observe very well a very strongly interacting species, but the interactions were not quantified. And because they were not quantified, that allowed people to come to very different conclusions about how to manage this species. Because some people thought, you know, the management threshold should be here, and other people thought it should be here. And some people thought this was going to be enough work on it, you know, to get it down to this level, and other people thought that would not be enough. And so the scientific quantification has actually been essential to resolving that management issue. Not resolved yet, but it's contributing. So Wes has been an amazing field scientist. He's been very efficient at writing. He has 2 chapters published, one that he just submitted. So that's 3 altogether. Check, check, check. Also, he is an effective code switcher in his ability to communicate across people from really different backgrounds. And I think that is a skill that really serves him well. And then he's also never lost his childhood joy of being out on the bay. And I appreciate that. So of the five major field experiments that Wes carried out, 4 of them were completely like demonic intervention. Something happened that he did not expect. And for those of you who do not know, one of those was that I can truly say about Wes that he is the only PhD student I know of whose experiment was eaten by a whale. And now I really hope you will do this with me. Please welcome Wes, not with laughter or clapping. Sorry, I'm clapping, but with the universal sign for crap. Oh yeah. I love it.
All right, that is not what I expected, but it was great. Please don't do that because I will laugh. All right, so thanks everyone for coming to my dissertation defense, and we're just going to get right into it. I think at this point you've probably read my title slide. Although certainly I can figure out how technology works. All right. Yeah, I guess. Oh. Let's see. Oh, this would happen to me. All right. Yeah, the point is on. Check that.
Maybe you're in the Zoom page instead of PowerPoint.
Oh.
Okay. Oh yeah.
Like we said, this isn't a PhD in a tech. Thanks to do. All right, so, and then let me just start it. Sorry. This is just really making it easier for me here. All right, so with that, before I start talking about my dissertation, first I want to go through a roadmap of how this is going to go down. So first I'll introduce the theory behind my dissertation, and then I'll introduce you to the system that I did the work in. I'll follow that up with a chapter overview. Then I'll go through each chapter one by one. I'll finish it with some concluding themes, and then I'll open it up to questions. So first, an introduction into that theory, namely what ecosystem engineers are and their interactions. So an ecosystem engineer is going to be an organism within its environment that's going to modulate the availability of some resource to other species by causing some sort of physical change in that system. This can manifest as a structural change, which can then result in either an abiotic or a biotic change. This can then create these feedbacks that help promote that engineer in that system and allow them to both grow and expand and create conditions that may cause self-facilitation of that engineer in that environment. So there are two types of engineers that I really want to focus on here, with the first being what's called an allogenic engineer. This is going to be an engineer that's going to transform the materials from one state to another within its environment, and it's going to do this mechanically. I think a good example of this in terrestrial realms or on land could be things like moles or gophers, things that burrow in the ground. And then we have autogenic engineers, which are going to be organisms that are going to manipulate that environment through the creation of some sort of structure within that system. This is going to be something like trees on land. Now, we're not going to talk about the land. We're going to talk about the aquatic environment. And so what we're looking at here is what a typical tide flat may look like here in Washington State. And what I want us to understand from looking at this picture is it's an important transitional zone in between the aquatic and the terrestrial environment. This system can support a wide array of different types of species, including those that can manipulate the function and the structure of these particular tide flat systems. And because of this, we actually have an opportunity to look at how these organisms interact with each other so we can see how these tide flats can change. So on tide flats, we have three main types of ecosystem engineers that we're going to focus on here. We have bioturbating species. These are going to be organisms that are going to be stirring up and manipulating the sediment that they're living in. This can cause that sediment to become destabilized and cast a lot more sediment into the water column. We then have organisms like seagrass and bivalves, which are going to be able to create that above ground structure that can then not only help stabilize those tide flats by causing sediments to settle out within their habitat, but also create some sort of habitat for other organisms as well within those systems. Now, the interactions between these three types of engineers have been extensively studied throughout the literature. And as you can see, based off the direction of the arrows, that who gains priority in these systems may depend on the context in which they're examined in. However, there are still aspects of these interactions that either haven't been looked at or are still poorly understood, and that could provide further explanation to these types of interactions. namely being the habitats that these organisms create. We can actually look at the distribution of these organisms across those borders to better understand how they're interacting with each other. Also, many of these organisms have unique life history stages that may actually matter in determining basically who gains priority on those tide flats, depending on what life history stage they're in. And then finally, where we can actually see who gains priority, it's not always clear whether or not the effects that cause that organism to be the more dominant in that system is occurring directly or indirectly through some other pathway, say the alteration of that environment that can create conditions that are less suitable for the less dominant organism. And so with that, I'd like to introduce you to the system in which I did my dissertation. These are the outer coast estuaries in Washington State, namely Grays Harbor and Willapa Bay. And in these systems, we have three common types of engineering species. We have an allogenic engineer, which is going to be burrowing shrimp, Neotripea californiensis here on the left. We then have autogenic engineers, eelgrass, Zostra marina, as well as an introduced oyster, the Pacific oyster, Magellana gigas. Now, interactions between these organisms have also been studied within these systems. However, these aspects of looking at their habitat borders and these different life history stages, as well as the direct and indirect pathways that may be influencing the distribution of these species on tide flats, have not really been considered for these species. And so with that, this brings me to my 3 chapters in my dissertation, with the first being titled Antagonistic Ecosystem Engineering Effects Differ by Seagrass Life History Stage and the Density of Burrowing Shrimp. So first, I want to provide you a little bit of context. So interactions between eelgrass and burrowing shrimp have been examined in California estuaries and bays in the past. And through transplantation experiments, by actually putting these organisms into one another's habitats, they've come to the conclusion that in those systems, eelgrass tend to be the more competitively dominant of the 2 species, likely through the formation of rhizomes that actually prevent those shrimp from being able to reach the surface. However, a lot of times when we're trying to get densities of organisms that live underground, we may utilize indirect measures of their density in order to infer what's happening with their densities. And additionally, this type of research has not considered the unique life history stages that eelgrass can have when it's existing within its environment, namely the early seedling stage, as well as an adult vegetative shoot stage. And then finally, as you can see in these images here on the right, we can see that there are differences or edges to the habitats that these organisms create, which could be useful for inferring these interactions and understanding more about how these organisms may interact with each other. So this has led me to 3 main questions for my first chapter. Or first, I examined how do the densities of shrimp relate to these abrupt habitat borders of eelgrass within these outer coast estuaries. So to answer this question, we identified 8 borders of eelgrass within Willapa Bay. And at each one of these borders, we measured the density of these organisms at distances away from that border on either side. And we got direct measures of burrowing shrimp using this clam gun coring method. And then we counted eelgrass and quadrats. And then we looked at the relationship of those species across those boundaries. And so now on to the results. So first, let's look at the graph on the left. So on the x-axis, we have the relative distance along those transects, with this red dashed line representing the center of the border. The green side represents the eelgrass side of the border, and the non-colored side represents the shrimp side of the border. And what we see right off the bat, at least for both shrimp density and shoot density, as they are not mirror images of one another, we actually see that eelgrass declines pretty abruptly across the border. This is likely an artifact of us identifying those eelgrass borders. But I think the interesting thing is here that when we look at the shrimp density, we actually see that not only is there a lot of shrimp just hanging out in the eelgrass, but their distribution or their densities actually increase as we travel across those borders. Additionally, if we look at the relationship between shrimp density on the X and shoot density on the Y, we can see as shrimp density increases, we see this decline in shoot density, and this value of around 330 shrimp per meter squared could represent sort of this shrimp density, beyond which we might not expect eelgrass to persist in these environments. All right, so now on to our second question, which is how do the effects of burrowing shrimp on eelgrass differ across shrimp densities? So to ask this question, what we did is we took 20 to 25 eelgrass shoots and we transplanted them into areas with varying levels of burrowing shrimp density throughout the outer coast estuaries. And then we just measured how long it took for those guys to survive at those different densities. And so now onto the results of this second question. So on the x-axis, we have shrimp density. On the y-axis, we have the proportion of surviving shoots within those transplant plots. And what we learned was that regardless of the region in which we did this in, and regardless of the time since transplantation, we learned that as shrimp density increases, we see a decline in shoot survival. With this 300 shrimp per meter squared mark is where we saw basically shoots only surviving for about a month. And now on to our last question for chapter 1, which is, how do the effects of shrimp on eelgrass differ between adult eelgrass and seedlings? So in order to answer this question, what we did is we transplanted both seedlings and vegetative shoots into an area of low shrimp density and high shrimp density. In each one of those locations, we then excluded shrimp from half of those plots by putting burlap 10 centimeters below the surface. And then we left the other plots the same. That way we could actually examine the effects of burrowing shrimp on the two different life history stages of eelgrass. Now, we allowed this experiment to run a couple months throughout the summer. However, we came back in the first 2 weeks to look at the survival of our two different eelgrass life history stages in the presence of shrimp. And so first, we're going to look at the results within the low shrimp density area. So on the x-axis, we have months. On the y-axis, we have the proportion of surviving shoots within those plots. Our right panel represents the shrimp exclusion plots, and our left panel represents the controls, where those life history stages were exposed to shrimp. And the shapes of the points represent the different life history stages. And what we learned is that there really wasn't an effect of shrimp exclusion at these lower densities. There was truly only an effect of the life history stage, in that vegetative shoots just tended to have a better survival than seedlings. And this is probably due to the fact that these are just bigger, more robust shoots. They're going to be able to stay anchored in the ground better than a seedling. And so at these lower shoot densities, or at these lower shrimp densities, what we would suggest is that there really isn't this life history stage effect that shrimp is having. Now let's get to the high shrimp density area. So these figures are formatted the exact same way, but we're just going to focus on the figures here on the bottom. And so if we first look within the exclusion plots here, what we can see is that when high densities of shrimp are actually excluded, we have pretty good survival of both seedlings and vegetative shoots. However, when they're exposed to the presence of those burrowing shrimp, not only do we see a decline in survival for both vegetative shoots and seedlings, but that negative effect is stronger on that early life seedling stage, suggesting that at higher densities, these burrowing shrimp are going to have a stronger negative effect on the early life history of eelgrass. And so what are my takeaways for chapter 1? Where shrimp are going to have a strong antagonistic engineering effect in the outer coast estuaries on eelgrass, where as their densities increase, we're actually going to see the preclusion of successful seedling establishment, as well as the preclusion of persistence of eelgrass, where shrimp may move into areas that have eelgrass. Additionally, these abrupt borders of eelgrass were not mirrored by shrimp. And as we saw, shrimp were never excluded across the full range of eelgrass densities that we sampled, which would suggest that rather than eelgrass being the more competitively dominant, or these sites being alternative to one another, shrimp are likely the antagonistic winner of these interactions within the outer coast estuaries. All right, so now on to chapter 2, which is titled Sediment Burial Caused by Bioturbating Shrimp Negatively Affect Juvenile Oyster Survival and Size. So I think we can all agree that if you're an organism like a little oyster that's living on a tide flat, you're probably going to be influenced by the sediment properties of that tide flat, namely how muddy it is and how soft or how penetrable it is. Additionally, you're going to be affected by organisms that are reworking that sediment in that environment, like burrowing shrimp. So therefore, burrowing shrimp may have two avenues by which they're going to affect small clusters of oysters on tide flats. Directly through burying these oysters in sediment as they eject sediment from their burrows, or indirectly by manipulating the sediment properties within that environment. However, which one of these mechanisms is really responsible for decline in oyster survival on these tide flats isn't clearly understood. And additionally, it's not totally understood how the sediment ejection rate of shrimp compares to just sediment deposition within a shrimp bed or in an area where burrowing shrimp have been removed. So with that, I have 4 main questions for chapter 2, with the first being how much of that total variation in sediment properties across these different estuarine sites may be related to the shrimp density at those sites. So to answer this question, we established 6 different locations within Willapa Bay, and then at each one of those locations, we established 5 to 6 sites. And at each one of these sites, we collected information on sediment grain size, sediment penetrability, and we got measures of shrimp density. We then examined the relationship between burrowing shrimp density and both our sediment grain size and our sediment penetrability. So let's look at the results from that. So what we're looking at here is a graph for a principal components analysis where the points represent individual sediment samples and the colors of the points represent the shrimp density associated with those samples. On the x-axis, this is a measure of muddiness within the sediment, where on the left side, we have less muddy sediments. On the right side, we have more muddy sediments. On the y-axis, this is a measure of sand grain size, or at the bottom, sand grain sizes are smaller, and at the top, sand grain sizes are larger. And what we learned was that burrowing shrimp are really only having a significant effect on the mud content within this sediment, but not the sand grain size. So as bioturbators, they're going to be reworking this sediment and kicking sediment out of their burrow. This is going to cause those fine particles or that mud to get kicked into the water column, where it's going to get transported to adjacent areas within the bed. And potentially over time, we could see this net loss of sediment or of mud as it gets kicked out of that environment. Sand grain size, though, it may be influenced by, say, larger, stronger forces within that environment, things like the hydrodynamics within that location or flow, whether it be seasonal or just in general in that site. And then additionally, what we learned was that as these shrimp densities increase, we see that those tide flats become softer or that penetrability increases, likely due to this sediment reworking effect. All right, so now on to question 2 and question 3, which are related to each other. So is oyster survival going to be better predicted by shrimp density directly or via indirect pathways through which these shrimp are manipulating sediment properties? And in instances where oysters are surviving on these tide flats, is oyster size going to be better predicted by that shrimp density, or is it going to be better predicted by those manipulated sediment properties? So to answer this question, what we did is we outplanted seeded culch at each one of these sites that we previously established. Now, what seeded culch is it's oyster shell that has had larval oysters settle on it. And then those turn into little tiny versions of oysters, and then they grow into clusters on that shell. This is a common method used for what's called bottom culture within the outer coast estuaries, and farmers will generally go out and then harvest these after a growing season. What we did is we went back out and harvested these at the end of a regular summer growing season, and then looked at how many survived on those shells and how large they got. So then what we ended up with was information on shrimp density, we ended up with information on sediment, and then we ended up with information on oyster survival that we used to construct a structural equation model that I'm going to break down into its components. So how you would read this is the direction of the arrow represents the direction of the effect, and the size of the value represents the size of the effect, and the asterisk is just denoting if that effect is significant. So how we would read this is increasing burrowing shrimp causes a decline in oyster survival. Increases in burrowing shrimp densities also causes an increase in sediment penetrability, and increases in sediment penetrability causes a decline in oyster survival. However, the effects of burrowing shrimp density on oyster survival mediated through its effects on sediment penetrability is not significant. So in this relationship, we would say that burrowing shrimp density is likely a good predictor of oyster survival on tide flats. Now, as burrowing shrimp densities increase, we also see a decline in the mud content within sediments, but the amount of mud that's in the sediment is really not having an effect on oyster survival, and neither is the effect of shrimp density on oyster survival mediated through their effect on sediment muddiness, further suggesting that this metric of shrimp density is just likely a good predictor of oyster survival on tide flats where shrimp are present. And if we look at this relationship a little bit more closely, on the x-axis, we have shrimp density. On the y-axis, we have the proportion of living oysters per shell. And what we can see is as that shrimp density increases, we see a pretty drastic decline in their survival, with this red area representing shrimp densities where we might expect, say, total failure of oysters on tide flats. And if you want to know what that means, it looks like this, where you have to dig your oysters out of like 40 centimeters of sediment because your shrimp buried it over 127 days. All right, so now what we're looking at here is the results of shrimp density effects on oyster size. And so these are the oysters that survived living in these shrimpy environments. And so what we learned through some model selection here was that the best predictor of our metrics of oyster size was just shrimp density alone, and the effect of shrimp density was significant for both shell height and tissue mass. Now, shell height and tissue mass were correlated with each other, so I'm only going to talk about shell height here in the graph. But what we see here is, of course, as our shrimp density increases, probably to no surprise, we see a decline in oyster shell height. So when we have these shrimp density increases, we get more dead oysters and smaller oysters. All right, so now on to our last question, which is, what is that sediment ejection rate of burrowing shrimp, and how does that relate to sediment deposition within a burrowing shrimp bed and in areas where they've been removed? So to answer this question, we ran this experiment within an active shrimp bed within Willapa Bay, where we took cotton cloth with a centralized hole and placed that over the opening of burrowing shrimp burrows. That way we could capture the amount of sediment that they're ejecting in a 24-hour period. We then replicated that within the shrimp bed, but between burrows, so we can understand how much sediment is getting deposited in that environment. And then we did this again in an area where shrimp were removed, and then we compared the sediment volume that was captured in those tubes over that 24-hour period. So now on to the results. So on the x-axis, we have our different treatments in the order that I explained them. On the y-axis, we have sediment volume. And what we can see is that we had a greater amount of sediment that was captured inside of tubes that were placed over those shrimp burrows. How much sediment was that? Well, it was about 29 millimeters or milliliters getting ejected from a burrow per day, which equals about 3 millimeters of sediment getting added to the surface of a tide flat within a meter squared area per day. If you multiply that out by the 127 days that we did our experiment, that equals about 38 centimeters of sediment that are getting kicked out and reprocessed through the sediment conveyor belt that shrimp is creating. And that's about as deep as our oysters got buried over the course of the experiment. So probably a pretty likely mechanism here. Additionally, what we're also seeing is that we just see greater sediment volumes within our tubes that were placed within the shrimp bed compared to areas where shrimp were removed. And this could be explained by the fact that we're just going to have more bioturbation in a shrimp bed. And so we're going to end up with more sediments that accumulate in those areas. All right, so on to our takeaways for chapter 2 here. So first off, shrimp are going to have an effect on the finer end of the sediment grain size scale, where they're kicking out those muds, and through time, through this sediment conveyor belt, they're going to kick that mud out, and we're going to end up with less muds within a shrimp bed. Additionally, we learned that shrimp density is just a good predictor of oyster size as well as survival, and this is important to know because this represents a metric that we can use as habitat managers and farmers to assess the quality of a shellfish bed, or just how oysters might be affected by shrimp densities within these systems. And then finally, we've pretty well identified this mechanism, or this direct mechanism of sediment burial that's causing the decline of oysters, where as these shrimp are kicking the sediment out, the heavier stuff lands on top, and over time we get these oysters that are buried that then eventually die. All right, so now on to my last chapter, which is titled Shoot Collection Intensity and Transplant Configuration Affects Intertidal Eelgrass Restoration Success. And so earlier I told you that eelgrass was an ecosystem engineer due to the way in which it's manipulating its physical environment, but it's also what's considered a foundation species, as it plays an important role in maintaining the structure of the ecosystems that it is in and those habitats that it creates. And it does this by providing habitat for forage fish and other local species within those systems, which inherently boosts biodiversity within that system. We also get an added benefit of improved water quality, as well as some carbon storage, as those particles are going to settle out in that bed, and then they're going to hopefully get stored there if we don't disturb it. And then finally, these presence of eelgrass beds are going to help prevent shoreline erosion and protect infrastructure along shorelines. It's also going to support different types of fisheries that are within that system by providing habitat for species that might utilize that space at some point in their life, or that are related to that environment in some indirect way. And so therefore, I think it's pretty easy to understand that we have a vested interest in ensuring that we make sure that these habitats are maintained within their environments, but also restoring them in locations where they've been lost. Now, sort of the general consensus of eelgrass restoration is that if you want to have a more successful transplant that takes root, you want to have a larger transplant that probably has denser shoot or more dense shoots within it, because what you might expect is that you're going to create these self-facilitating conditions that are going to promote the growth and survival of that eelgrass. Additionally, when you're sourcing from these donor sites to get that eelgrass, you want to collect from locations that are big, healthy beds. That way you can mitigate the effect of collection on those source locations. That way you're not ruining eelgrass when you're trying to transplant it. However, in stressful intertidal areas, right, eelgrass can occur as these sort of sparsely populated fringing beds at the lower end of that intertidal elevation, but also as these sparsely populated beds at the upper portion of the intertidal, which sort of calls into question basically the effects of collection intensity on these types of eelgrass populations. And additionally, this question of how plot size and both plot density for eelgrass affects the restoration success in these environments is poorly understood. And so this leads me to three main questions for my third chapter, with the first being, how rapidly do different levels of shoot collection return to background densities at these lower and higher intertidal eelgrass beds? So in order to answer this question, we identified two intertidal eelgrass beds within Willapa Bay, one at a higher elevation and one at a lower elevation. And then we established 4 different collection treatments at both of those sites. The first, which isn't pictured here, was a reference treatment, which was just eelgrass that had its plot boundaries marked, but we didn't remove anything. We then created a 50% collection, an 80% collection, and 100% collection treatment. And we then compared how long it took for these different treatments to revert back to similar densities of those reference plots at these 2 locations. And we tracked this over the course of 4 years. All right, so now on to the results. So on the x-axis, what we have here is time in days. On the left y-axis, we have shoot density. On the right y-axis, we have shrimp density, because we're not done talking about shrimp. And then each one of these dashed red lines represents the point at which I analyzed my data. I've split these graphs up into the low elevation site and the high elevation site. And then the colors represent the collection intensities, with the exception of the black lines, which represent shrimp density. And so through this experiment, what we learned was that regardless of elevation, lower collection intensities are just going to lead to faster recovery at these 2 different elevations. Additionally, what we learned was that generally speaking, these lower elevations had higher densities than our higher elevation site. And this could be explained to some different sources of disturbance that we may have witnessed at that particular site. Namely, if we look at the shrimp densities at these sites, we know that shrimp negatively affect eelgrass. And so when we have these drastic increases followed by declines of shrimp, we may expect large fluctuations in actual eelgrass density in those sites. However, the thing that's really important to note is when we actually compare the recovery rates of our most intense treatment here, our 100% removals, they're in keeping with the recovery rate of other eelgrass collection intensity experiments that have been done in Willapa Bay in these larger, healthier beds, which could suggest that these sparsely populated areas are going to recover at a similar rate as these otherwise larger, healthier beds. All right, so now on to our second question for chapter 3, which addresses how does transplant density and plot size affect A, the initial shoot persistence after 2 months, B, infilling within those plots, and C, population growth. So in order to do this experiment, all of the shoots that we collected from our collection intensity experiment, plus some extras, we used to create this transplant experiment where we crossed 4 different plot sizes with 3 different transplant densities. We used eelgrass from both donor sites and created these large blocks that we then replicated within the intertidal area to create these 10 large blocks that contain all of those treatment combinations. And what that results in is an area where you transplant roughly 10,000 shoots into a transplant area that you then monitor over the course of 2 years. Now, additionally, it's important to note that at the time of this transplantation, this was also an active shrimp bed because we wanted to know how these different transplant configurations affected shrimp and vice versa. but by the spring of the following year, the majority of all that shrimp were no longer present in that habitat. All right, so now on to the results. So first, we're going to look at the shoot retention within these plots within the first 2 months. So on the x-axis, we have the initial transplanted shoot density, and on the y-axis, we have the proportion of shoots remaining in those treatments, and then the colors represent the plot size. And what we learned was that for our tiniest plots here in pink, the initial transplant density really had no effect on shoot retention. However, we did see an effect for our other larger plot sizes. And we actually saw what you might expect to be the opposite, given that we would have otherwise expect self-facilitation to occur when we have higher eelgrass densities. What we actually see here is as that transplant density increases on the X, we see a decline in shoot retention on the Y, which could suggest that perhaps we're seeing some intraspecific competition among shoots here within these plots. All right, so now on to the results of our plot infilling, which I have defined just as eelgrass expanding to fill the gaps within those plots, right? So similar sort of format here. On the x-axis, we have transplant shoot density, and then on our 2 y-axes, we have shoot density. I've split this up into 2021 and 2022 because these are the 2 years that we analyzed this data, and I have plot size represented by the color. You might notice that our tiny plots are missing. That's because the eelgrass in those tiny little plots migrated outside of their plot area, and so therefore I couldn't actually measure it. And so let's look at 2021. So what we discovered was that within that year between 2020 and 2021, large and medium plot size filled all the way in and converged to similar shoot densities. However, these smaller plot sizes, what we see is as transplant density increases, we see an increase in shoot density. And what could be going on here in 2021 is really that these more sparsely populated plots just have more room for that eelgrass to fill in and expand versus these larger plots. And so we'd expect infilling to just occur faster regardless of density in that case. Now in 2022, what we observed is a whole scale increase in just the shoot densities within these plots, but these relationships remained pretty much the same. All right, so now on to our last set of results for our second question, which examines population growth. So on the x-axis, we have time in days. On the y-axis, we have the proportional change in total shoots. Each panel represents a different plot size, with our smallest in the top left and our largest in the bottom right. And then the color again, or the color now, represents our initial transplant densities. And again, we examined it both in 2021 and in 2022. And so what we see in 2021 is that these smaller plot sizes with less dense shoots had the greatest increase in the amount of shoots within those plots, which would suggest that they're experiencing the most rapid expansion compared to their larger, more dense counterparts. In 2022, we see the strength of that relationship increase, where we really see these smaller or these tiny, less dense plots really taking off. And you can really see this when you actually examine the scale of the y-axis compared to the rest, right? One thing that's also important to note here, which is why I circled this particular point, is we have further evidence to suggest that expansion is occurring really rapidly in these plots. So what I'm not showing you here is the results of shoot flowering within these plots. But what we know about eelgrass in this system is that within the first year of a shoot emerging, it's not going to be able to flower within that first year. And we saw the lowest proportion of flowering shoots in our tiny, most sparsely populated plots, which suggests that we have this really rapid expansion that's creating a lot of these fresh new shoots. And so therefore, you just can't have those shoots flower. And so otherwise, you would see all these other plots, these larger, more dense plots, having higher proportions of flowering shoots, which just reinforces that these tiny, less dense plots are performing really well. All right, and so now on to our last question here, which addresses, are essential services like sediment deposition achieved through eelgrass transplantation? Because part of the goal with restoration is to not only bring the eelgrass back, but to get some of those services back, right? And so the way in which we examined this is we simply collected sediment samples from inside and outside of our largest, most dense eelgrass patches, and then looked at the proportion of fine particles and organics between those between those plots and areas where there was no eelgrass through time. And so on the x-axis, we have time once again. On the two left axes, we have the percent of fine particles and organics within our sediments. And on the two right axes, we have shrimp density. Again, along the top are the points in which we analyze the data, and now the color represents whether or not the sample was collected inside of eelgrass or outside of eelgrass. And so let's first look at the results for 2021. And what we saw was, as expected, when you transplant eelgrass into an area that otherwise is void of some sort of above ground structure, we see higher fines and organics within those environments, suggesting that perhaps we're getting some of that service occurring, right? However, what we see in 2022 is not only just a whole scale increase in both fines and organics inside and outside of eelgrass, but that relationship actually flips. And what could be going on here is two things. One, we know that when you put eelgrass in these plots into areas that otherwise are void of above ground structure, we can actually end up getting sediment scour and sediment ascension in and around those plots, which can actually cause those fines to get moved out of those plots, resulting in greater fines and organics around those plots. So that's one thing that could be occurring. The other thing that also could be happening here is, you notice we see these black lines, these represent shrimp. We see a pretty drastic decline in shrimp density in these plots. And so what that means is we're losing that bioturbating activity of shrimp within this area, which suggests that rather than muds getting kicked out, the sort of overarching normal sediment deposition that could be occurring in that environment is now occurring because the shrimp isn't there. And it could just be happening at a faster rate than, say, in the eelgrass, likely also due to the first reason that I mentioned as well. Then what we see for 2022 is, regardless of that difference, we see the convergence of fines and organics between inside and outside of those plots. And what may be explaining this is, by the second year, the eelgrass pretty much filled in most of this entire area. And so we're just seeing that whole scale establishment of that service within that area. All right, so now onto our takeaways for my final chapter, which is one, low intensity sheet collection is going to ensure more rapid eelgrass recovery. I think that's a no-brainer, but I think the important thing to note here is that this recovery for these lower fringing and upper margin sparsely populated eelgrass beds can recover at a similar rate as these larger, healthier beds within that same system, which could qualify them as potentially good sources to use for eelgrass restoration. We also saw little evidence of self-facilitation in eelgrass transplants, and more evidence of intraspecific competition, where the shoots may actually be detrimental to each other and competing with space, and actually having smaller plots with less shoots is allowing for them to grow and expand at a more rapid rate. And then finally, we did see the expected service of sediment deposition occur within these environments due to the transplantation of eelgrass. However, the rate at which that may occur could depend on biological changes in those environments, like shrimp leaving that area and then, say, abiotic conditions then coming in, but also just the nature of the transplantation itself. All right, so now on to my overall conclusions and themes for my dissertation. So first, borders between engineering habitats are really useful for inferring interactions between these different engineering species, and the effects of these antagonistic interactions can depend on the life history stage at which they occur, where we could see that perhaps at early life history stages, we may see stronger effects on those particular organisms. Now, this is really important to know because it helps improve our understanding about not only interactions between these tide flight engineers, but it also helps us better understand and also identify important factors that may be contributing to who gains priority, but also what those potential services and functions are in that system based off of those interactions. We also learned that burrowing shrimp density is likely a really good predictor of oyster performance, probably also eelgrass density. And this is valuable from a management perspective because it provides us with a metric that we can use that we can actually go out and measure pretty easily. Additionally, the sediment burial, which we are calling here that direct mechanism that's causing the decline of oysters, ties into that burrowing shrimp density really nicely, because now not only do we have a tool that we can use, but we also have a mechanism that can be attributed to what we're seeing out in these areas, which from a management perspective, as a shellfish farmer, or even as a management or a habitat manager from an agency-wide level, this is the types of things we want to know when other things may not explain why we're seeing changes in these systems. And then finally, in regards to eelgrass restoration, like what I previously said, lower collection intensity is going to ensure faster donor site recovery, especially in these fringing and upper margin eelgrass habitats. And from an ecological standpoint, it might be the best bang for our buck to actually consider smaller transplantations with lower densities of shoots, because it could ensure the more rapid establishment and expansion of these eelgrass plots. And from the economic standpoint of restoration, because restoration costs money, we're always looking for ways to not only improve our restoration outcomes, but to make sure that we can continue to do restoration in a manner that isn't necessarily going to break the bank. All right, and so there's like an enormous amount of people that I need to thank for all this work. First, I'm going to thank all the farmers that I interacted with over the course of this work. Without getting the ability to walk all over their tide flats and like break all our backs, scoring shrimp and crawling around on the ground, pulling out eelgrass, I wouldn't have been able to do this work, right? And so I think what this really shows is that farmers do really have a vested interest in the research that we do out in these estuaries. And like us, they also understand the ecological value of getting this type of information, because not only does it make us better at understanding these processes, but it also helps them be better stewards of their environments while also being more profitable. So I just want to say thank you to them. I also want to thank my funding sources here at the bottom. So that's like the Department of Ag, Fish and Wildlife, PSI, Sea Grant for providing funding for this project. I also want to thank the Department of Natural Resources, who I work for letting me continue to work on this project. I think that Washington State is a really unique sort of scenario where you have all these agencies that really have vested interest in work that we do here and work that's happening on the ground. And I really just think that we have a really good outlook going forward in regards to just being good stewards of our environment, knowing that these departments are willing to work with us to try and answer these questions that sort of transcend ecology in both management and production, right? Money making, right? And then finally, it really takes an army to get a dissertation done. So I don't want anybody to think that I did most of this work, because I certainly did not. I could not get it done without many people who are in this room. And so when I think about this work, I don't think of it as just mine, but I kind of think about it as ours, because all of you had a small or sometimes large role in making sure that I was able to get this done. And so for that, I want to thank you guys. I'm not volunteering to help you with your field work necessarily, but if you for some reason ask me to, I might say yes, because I might feel obligated. But again, thank you for that help. I do like going out in the field and poking stuff. I also need to thank all my friends and family that have been with me throughout this entire process. Sometimes I think I'm like a student who's been in college for like way too long. I think it's been like 15 years since I started college, and here I am. And there's been lots of people that have been there through that entire process that have supported me, that have helped me get through the hard times, that have provided fun times, provided confusing times. Sometimes they were just there and I just needed someone to talk at or yell at, or I don't know, just stare at, because I didn't know what was going on. So I want to say thank you. That unwavering support, while I may not express it very often, I really value. And so thank you for that. There's a couple of people that I have to thank specifically for that support. So the first being my fiance, Meg. She's the one with this foam finger in the audience. So I've been with Meg for like 18 years, right? See, it's a good thing I'm not going to do a dissertation on that. Meg's been there through every step of the way. She's watched me go on these huge, endeavors of trying to sample at early morning tides, coming home, being tired, being confused, not knowing what's going on, and then having to deal with like a mopey boyfriend being like, man, maybe ecology isn't for me. That's not easy to do. And I realize that I also don't make it easier, because I can be a little bit of a drama queen behind closed doors. Additionally, I think throughout that process, Meg has just shown unconditional love and support that I don't know if I can really replicate, because it truly is amazing. And so for that, I want to thank you. And I promise I'm not going to talk about shrimp at least till the end of this week. And then the last person that I really need to thank is my advisor, Jen. So like Jen said, we met in 2020 prior to the pandemic, and I asked her if I can be in her lab. That manifested into me being a technician for a year. And then that resulted in me being a part of this awesome community that is currently watching me fumble my words up here currently. I think Jen is a really good example of just sort of what the full package can be, right? I think that from an academic standpoint, I had an opportunity to observe someone that represents an exceptional role model for how to conduct yourself as a scientist, both in the field, but just when you interact with your, your counterparts in these systems. And that's something that I will always draw on as just sort of advice and something to think back to as I work through my professional career. Additionally, I think Jen walks that line between being a scientist, being a mentor, and just being a person that is a part of the community so well, better than other people that have had that role in my life previously. It is not easy to be a scientist and convey information to groups that otherwise might view you as someone who's going to prevent them from doing something. I think Jen has this superpower to make friends with everybody and communicate that information in a way that where everybody values from it. So moving forward in my career, while my academic career is probably ending now, hopefully, I'm certainly not coming back to get another PhD. I will remember all of those experiences I've had with you, and I will draw on those. And then moving forward, while we may not work in this setting together anymore, I certainly look forward to working with you as we move forward into the future from a professional standpoint. So with that, I'll open it up to any questions. My mouth is dry. Oh God. I'll do Madeline because she raised it first.
Really great talk. It was super interesting. I was actually really curious about, thinking about eelgrass density. So you talked about how when you have basically fewer shoots transplanted, they have the ability to sort of burst through that growth and you see a higher % change in growth overall. This probably wasn't the aim of your dissertation at all, but did you see an impact of given how density of eelgrass in an area, how that impacts shrimp survival? So sort of looking at the reverse relationship and whether or not you have more eelgrass, are you less likely to have shrimp there? And yeah, so sort of thinking about that other side of the arrow of how eelgrass density impacts shrimp survival, and if you noticed anything.
Yeah. Yeah, certainly. So at least with the transplant, initially that was the goal, was to sort of look at that relationship between eelgrass transplant size and density and their effects on shrimp. Unfortunately, like Jen mentioned, I got part of my chapter ruined by a whale, but also some of it also got ruined by shrimp because they leave those areas and I can't just I don't have a whistle to call them back. But from our border analysis that we did where we sampled across those, we do see that at least on the higher end of I'll say that there could be an effect of eelgrass density on shrimp. In these outer coast estuaries, what I'll say is the densities probably don't get high enough for shoots to actually purely kick eelgrass out. However, we do know that in different areas of Washington State, namely the Puget Sound, we can get eelgrass density that's a lot higher. And so I would expect that at certain higher densities, we would see probably a negative effect of burrowing shrimp. However, we haven't looked at that yet. But certainly in that example that I showed from California, those densities of eelgrass shoots are much higher than what we observed. I think, really high density for us in the outer coast estuaries is really going to be hovering around probably 100 shoots per meter squared, maybe a little bit more. In those California estuaries, we're talking 250, 300 shoots. And that's really making this dense network beneath the surface, which is probably that mechanism that's preventing those shrimp from getting to the surface because they have to get to the surface to do their things. So, but yeah, certainly, but at least in the outer coast estuaries, probably not. Yep.
Wes, congratulations on talking your thesis. Congratulations.
Thanks.
I'm pretty sure I am not the only one curious in this room, but what's the whale story that?
Oh, certainly. Yeah, I didn't actually think anybody was going to ask about the whale, but yeah, so my first chapter, what we were initially going to do is we actually went out and we created these plots and these 2 shrimp beds. And we pumped shrimp out using these water pumps that just, once you liquefy the sediment, the shrimp just come to the surface and then you can spray them off the bed and the seagulls eat them, right? So you get these nice areas where there's no shrimp inside of, these shrimp beds, right? And we were putting flowering shoots and anchoring them and putting these seed bags in that area so we could understand how burrowing shrimp would affect that seed stage of eelgrass. If those of you, I mean, hopefully some of you know about gray whale feeding behavior, but we have gray whales that travel up here and actually consume burrowing shrimp. And what happened was probably a day or two before we went out there, it looks like what had happened is some gray whales came in and just fed through that area. And so when we went out there, rather than seeing our plots, what we saw were these big giant pits of holes that had little like shrimp parts and stuff in them, which also mean we didn't have any plots anymore. So that's the whale story. You know, it's like the dog ate my homework, but like in this case, it's a whale ate my first chapter. So, and it's a real story. It's like true. I have pictures. Jen was there. Yeah, like I'm not lying to you. So that's the whale story. That's why it was also in my acknowledgments, because it was a character building exercise. This is what are you gonna do, right? We can't hunt whales anymore. So, Chris.
Awesome job. This was so cool to see all of the work. Congratulations. I had a question about the restoration work, the lower density shoots, and like that growth looked like it was starting to just get exponential at this point. Do you have any sense, like, is it gonna hit some sort of carrying capacity and then maybe crash back down, or is it like, does it just not matter up there? It's just gonna keep extending it?
Yeah, so certainly when you do eelgrass transplantation, when you do plant them at different densities, what can happen is you can get sort of self-thinning within those plots where you may end up with just sort of the density that eelgrass wants to be at. Certainly with those tiny plots, that would happen eventually, but I think at that point in time, when we are talking about the expansion of those plots, they haven't quite reached those densities within that area. They also had just a larger area to spread out into. So as I would expect that as that transplant sort of filled in all the gaps, we would probably see overall just sort of maybe these eelgrass sort of reaching a density at which they all sort of want to be at. But yeah, for the tiny plots, certainly I don't think so. Yeah, so I mean they were this big, so yeah, they're pretty small.
Cool, thank you.
Yep. Yeah.
Did a beautiful job of articulating what you did and what it means to get anything, but fumble for words as you said, about a beautiful presentation. On the theory side of things, in the background, keystone species, the ecosystem engineers are great concept that have been applied carefully initially. And then there's investigators and continuing. Everybody wants species they study to be everybody wants their species to be ecosystem.
Right.
You completely justified that. But as you study the system, do you see any other species in there that are ecosystem engineers and they have a big role and you have to study them? Or some species that are definitely not many things that you saw there besides those who didn't show up? Who else is it? Under the whale.
Yeah, right. Yeah, so I wouldn't say that we saw necessarily like hard evidence of other organisms coming in and showing like their ecosystem engineering potential at the same sort of magnitude as these guys. But we do know that in tide flat systems and other parts of the world where eelgrass is, there are things like large worm species that live within the environment that can create considerable amounts of bioturbation in those systems as well. And so while they're sort of having a similar mechanism, they are a different species. And so worms could potentially be that case. Now, like I said, we didn't see like a whole scale increase in like large burrowing worms in this environment, but we did see in some of our plots or in failed plots, right, that we sort of had to abandon, that we did see worms move in, different species of worms and these small little tube building worms, right, which certainly can have an effect on how sediment and how flow moves through that environment at that really close sort of like sediment water interface. But that would be my sort of first take is like certainly worms, and there are big worms out there as well. And so that would be one. As far as like
You just answer in terms of if you could do it again, what would you study about crabs?
Sure.
Favorite.
Yeah, sure. So,
favorite organism.
yeah, so if I were to study it again and what I would say or try and examine about crabs,
Are they important there?
crabs,
Crabs, are they important in the bay? And you didn't talk about it.
right? I didn't, which is probably good.
Yeah.
Yeah, certainly. So I don't think that crabs are going to necessarily have an engineering effect similar to these two species. Although what I might suggest just from some of the previous work that I did for my master's is I do think that crabs can have a pretty strong effect on those interactions that can occur within their environment, namely things like how they interact with, say, shared prey sources, or just environments, say, like higher up within sort of that estuarine system that they may be utilizing as habitat. So we do have hairy shore crabs that exist in these environments that are going to burrow into banks and stuff along those edges of those tide flats. Certainly, they're going to be manipulating the conditions within that, we'll say arena of sediment. And then certainly a lot of us probably know about the green crab trapping that's been happening in the outer coast estuaries as well as in the sound. And we already know that from that standpoint with that introduced species that they certainly can have strong effects within the environments that they end up occupying. And so there's been active work done by Sea Grant and other groups like WDFW to sort of look at what the potential effects of those are in those systems. And so while I do believe that crabs could serve an important role in that environment and affect how these habitats could be structured, me personally, I don't think they're going to be doing it in the same way as, say, our ecosystem engineers here, where they're either creating structure or they're really sort of stirring up that environment. Unless we're talking like little tiny, absolute huge populations of like shore crabs that are living sort of at that higher elevation. I don't know. Honestly, that's something I want to look at, but I haven't really convinced anybody to allow me to look at that with money yet. So, but maybe that'll be like my retirement goal. Who knows?
In Wilton Bay, I know that there's a bunch of new species that do look back a lot of oyster growers, like the green crab, also the oyster drill. So a question I have for you is when you're talking in your thesis, when you're talking about using seagrass, and seagrass can sometimes affect the density of burrowing shrimp. In your conversations with farmers and growers, has there been any interest in potentially increasing seagrass densities within their own beds to kind of deter burrowing shrimp populations?
That's a good question. I mean, historically, farmers used to be able to spray to eliminate shrimp on their beds, which was pretty effective. They don't do that anymore. And then previous work that our lab has participated in has looked at how mechanical control can sort of affect shrimp densities as well. And I think sort of the gist of that is mechanical control could work, but you got to put a lot of time and investment in doing it in order to make it work. And then there's still probably not a guarantee necessarily that shrimp might not invade from other areas. As far as using eelgrass as a restoration tool in aquaculture in the outer coast estuaries, I mean, certainly eelgrass grows in those aquaculture beds, and eelgrass is also a protected species because the value of that habitat that it creates is useful for sort of everybody utilizing that system and elsewhere. But in my experience, I haven't had those types of conversations with farmers on using eelgrass as a tool. And to be honest, I might expect that eelgrass can sometimes maybe represent just sort of an additional challenge when you're farming in those systems, because it's something that you're going to come across. You may uproot it. You're not, and you don't want to remove it, right, as well. And so, yeah, I'm not sure. I mean, the other thing that farmers have really, sort of used in the past to help mitigate the effects of shrimp is to really sort of just disturb that environment where a shrimp could be by either dredging it or harrowing it in some way where you're raking that ground, or even doing that to pull your oysters up to the surface, which they'll do probably regardless of the presence of shrimp or not. And so those are some tools that have been used in the past to also try and help with that. But yeah, I can't say that I really have had those like sit down conversations about like, hey, we thought about sticking our old pal eelgrass in your bottom culture bed, because they're going to come through and dredge that, right? And so otherwise they'd be transplanting eelgrass all the time, right? And they'd have to do it at a really high density. And in these outer coast estuaries, they might just get to a density to where it doesn't matter and the shrimp are still going to be there, right? And so maybe the shrimp just kick it out. So yeah, that's kind of my sort of answer to your question. Yeah. All right. Anybody else? Okay.
Okay,
I'm gonna end
so yes, let us thank Wes for participating. There's late lunch stuff outside, but the committee and Wes will stay here. And Haley's and lab members are in charge, but if you would like to join in on a burrito, especially friends and family, please do. And Wes, if you need a quick break,
Yep.
good, then come on back and we'll quiz you for the last little bit.
Great, thank you.